Hello, I'm Jody Rosenblatt from the Huntsman Cancer Institute at the University of Utah. And what we'll be talking about in this final segment is how epithelial cells maintain constant cell numbers through a process of cell extrusion and different diseases that can result if this extrusion process is disrupted. Now, in the previous segments, we identified different mechanical-based mechanisms for controlling epithelial cell numbers so that they maintain this constant density that's optimal for their function as a barrier. If the cells become too sparse, they'll activate cells to divide by sensing cells stretch to get back to these numbers. And if they become too crowded, they'll activate cells to extrude to get rid of these cells to go back to these densities. Now, if these processes are really critical for maintaining these constant numbers, you could imagine that different diseases could result if this was not maintained. And in fact, if you had too many cells extruding or too many cells dying, then you could get barrier function problems with this epithelium, and this could cause chronic inflammation. Furthermore, we'll see later in this talk that disrupting this extrusion process can cause cells to accumulate into these masses that form tumors, and also can lead to a new mechanism for how cells can invade called basal extrusion. So first, I'll tell you about a disease, asthma, which has really been thought of as an immune disease, but what we think may be really a result from poor barrier function due to too much extrusion. And the reason for this is really when you think about how an asthma attack occurs, it really comes from constriction, which cramps down in these airways. So normally, when cells are turning over in a normal, non-pathological state, it only takes 1.6-fold amount of crowding to cause some cells to extrude to keep constant cell numbers. Now, what would happen in this case where the airways clamp down through smooth muscle contraction, which contracts this airway and the epithelium here? This would experience so much crowding that we might expect to have excessive amounts of extrusion, which could just denude that whole epithelium and strip off the function that is the barrier here. And this could cause this inflammation that we would often see that's really thought of what asthma is, an immune disease. Now, this fits quite well with the chronology of symptoms that occur, is that most people, when they have an asthma attack, will notice that they have an inflammation for weeks or months following this. And at this point, they're really much more susceptible to other infections, upper airway infections from viruses and bacteria. And the problem with this is that this causes more inflammation and can lead to more attacks, and then you can get this whole inflammatory cycle. So we wanted to investigate this model of whether too much extrusion during a bronchoconstrictive event could cause this destruction of the airway epithelium. And to do that, we needed a system to look at this live. And so we developed an ex vivo lung slice contraction experiment to follow this. And what we did then was take some mice and immunoprime them with ovalbumin. And this step is not very well understood, except that it will cause an allergic response. And this did after several injections. We get the mouse become immunoprimed. Once this happens, we'll sacrifice the mice, harvest the lungs, and slice them. And then we can culture them in cell media for up to a week. At this point, we can test this idea of bronchoconstriction by triggering the cells, these bronchioles, to contract using a mimic of acetylcholine methacholine, and that will activate the smooth muscle contraction that causes this bronchoconstriction. So then you can see this through live imaging, and you can see this is a bronchiole right here, and as we add methacholine, this bronchiole will constrict. Now, if you zoom in a little bit and follow another bronchiole, you can see that a lot of these cells are already starting to extrude quite rapidly. So depending on the amounts of methacholine we would add or the amounts of priming, you can get subtle amounts of constriction or very extreme amounts of constriction where we could actually see these sort of involuting and cording because they're constricted so far. And we weren't really sure why they would respond in this way, But what we found was that the immunopriming was really important for driving one thing, and that was amplification of that smooth muscle. So as I said before, the smooth muscle surrounds the whole epithelium in the bronchiole. And before priming, you can see smooth muscle, but it's not very amplified. But after this priming with ovalbumin, it becomes greatly amplified. And what we notice is that when the smooth muscle wasn't very amplified, then it's not very reactive to methacholine. Whereas once we had primed it and it really became amplified, then it was very reactive to methacholine. So this suggests that maybe this is why some people respond to different stimulus by bronchoconstricting or not, and why people might be asthmatic or not. And so this is an interesting side product of being immunoprimed. So then what happens when we do these bronchoconstrictions? Well, if we stain the epithelia by staining for E-cadherin in these slices that are fixed, you can see the epithelium, even with small amounts of bronchoconstriction, there's still quite a few epithelia that are extruding. But in the cases where we saw extreme levels of constriction, there was so much extrusion or so many cells falling off into this airway that there was just denuding of that whole airway here. So you can see how this could be a real problem for an asthmatic. If there was this much cells in the airways, it would be hard to breathe after this. But also, there's very poor barrier function here, and you can see how it would be much easier for any kind of pathogens to get into the airways, and this could maybe aggravate this and cause more amplification of this problem. And so based on the fact that we knew that crowding-induced extrusion was controlled through piezo-1, a stretch-activated channel, we wondered if we could use a very common basic inhibitor of stretch-activated channels, which is gadolinium. And the reason we were interested in gadolinium is that it's already been used frequently in MRIs as a contrast dye that many people have used, and so we don't think it should have ill effects on the body. It seems to be well tolerated. And so here now we're going to constrict these bronchioles with gadolinium. And when we do that, you can see that this really prevents that extrusion and the falling in of this whole epithelium. You can see how nicely that they remain. And this was borne out in multiple different experiments that addition of gadolinium could block all these levels of extrusion, and even to these high amounts where almost the whole epithelium became stripped off that airway. So now we're really hoping that we can add gadolinium into inhalers and block that inflammatory period that occurs after an attack. And hopefully, by doing that, it would not only stop that inflammatory period after attack, but maybe block that whole inflammatory cycle that would lead to more attacks. Now, in addition to telling you about how disrupting extrusion can cause barrier function problems, we'll also look at how when you disrupt the signaling that drives cell extrusion can also cause tumors to form. And to look at this, we're going to look in our in vivo model system, which is Zebrafish Epidermis. And the reason we really like this is because it's very easy to see. If you look in the fin tail here, you can see it's just pure epithelium, a bilayer of epithelium. And that bilayer is very similar to the types of epithelia that coat many of our organs, but we can just readily film it. Okay, so now when we looked in a mutant that lacks the sphingosine phosphate signaling that's required for the extrusion, we found that extrusion was not only blocked in these zebrafish, but that also masses formed, much like the piezo result, but much larger masses formed at places where the cells should have extruded, compared to control wild-type zebrafish, which maintained a constant epithelium. We also found the same kind of result when we knocked down sphingosine phosphate 2 by shRNA-mediated knockdown. These cells also formed big masses and did not continue to grow in a monolayer like the control. And these masses were also resistant to apoptotic stimuli and chemotherapies. So they seem to have very high survival signaling. So while this idea that extrusion could drive cell death to maintain constant numbers, this seemed to really support that idea. But it also made us wonder, whether extrusion and sphingosine phosphate signaling could act as a tumor suppressor to prevent tumors from forming. And so to look at this, we went looking in the databases online to test whether this sphingosine phosphate pathway may be downregulated in different types of cancers. And what we found is that there was a particular type of aggressive cancer, it was pancreatic cancer, that had very little sphingosine phosphate 2 mRNA. We also found there were types of lung and colon cancers that also had reduced sphingosine phosphate 2 mRNA. But to test this, we really needed to look in tissue. Many people say the issue is the tissue when it comes to cancers. And so here we're looking at the epithelial cells in green. Now that's important because that's where the cancer originates again, is in epithelial cells. And if you look at the sphingosine phosphate 2 staining in red, if I take off the green channel, you can see that in the carcinoma there's really very little at all compared to the control uninvolved tissue here. Now precursor lesions called pannins had sort of intermediate values of sphingosine phosphate 2 staining. This was also seen in multiple different samples and also in matched samples from the same patient as this cancer progressed. You can see that the sphingosine phosphate 2 was gone. So this was an interesting correlation that sphingosine phosphate 2 may be playing an important role in cancer because it was lost in cancer. But we needed a way to test whether it really had a role. And so to do this, we started looking at a pancreatic cancer cell line. And this is called HPAF2, which also lacks the sphingosine phosphate 2 protein. As we saw before in the knockdown, these cells were unable to extrude, and they formed these masses as seen before. So it seemed that they were behaving very similar to the cells that lack sphingosine phosphate 2. Now, is the sphingosine phosphate 2 really important for this behavior? Well, what we did was test this in a mouse model. So we injected these cells into orthotopically into the pancreas of nude mice. And you can see that if you label them in GFP, that they form these huge masses throughout the whole body. This just shows you the whole mouse body cavity here. And you can see that it's really, the tumor is filling up the whole space, and there's lots of metastases occurring with this pancreatic cancer. Now, if you rescue sphingosine phosphate 2 tagged to GFP, you can see that this tumor is greatly reduced in its ability to grow, and there's also very few metastases. Only 1 out of 10 actually metastasize. So this really suggests that sphingosine phosphate 2 is important for this tumor's growth and metastasis. So one thing we would really like following these results is if we could rescue the sphingosine phosphate 2 in these tumors. But this seems very unlikely to target only cancer cells and put in a protein. And so what we wondered is whether we could do this with chemicals. And this is very important because this is a very aggressive sort of cancer where most people that get this will go on to die from it within 6 months. So anything we can do to help to fight this cancer would be fantastic. So we wondered whether we could actually chemically bypass the extrusion to trigger cells to die even when extrusion is blocked, as we've seen in these cases where the sphingosine phosphate is absent. And one good candidate is a key survival signaling focal adhesion kinase, which you can see is quite high in cells early on before they extrude. But later on, after they've extruded, this becomes greatly reduced. And this is due to the survival signaling that really activates this from being attached to the matrix and the surrounding cells. And so as the cell leaves, it loses that survival signaling, focal adhesion kinase. So what we wondered, in the case where extrusion is blocked, this survival signaling is prolonged, it stays there. Could we just block the focal adhesion kinase using an inhibitor and cause these cells to go on to die? And the answer appeared to be yes, we could. So now you can see this reduced rate of cell death in the control when extrusion is blocked. But if we add in focal adhesion kinase inhibitors, we rescue that amount of death and restore the rates of apoptosis to the levels of wild type. Now, a really fantastic thing we saw as a side part of this is that addition of focal adhesion kinase just to normal wild-type tissue had no impact on the cell's survival or death. And this is important because this is what we're always looking for in cancer drugs, is a therapeutic window where we're treating only the cells affected that are causing the tumor, but we don't affect the surrounding normal tissue. This is really critical when you see people who have been treated for cancer, because sometimes the treatment can be worse than the actual disease. It can cause cell death to your gut epithelium, which can make you really ill, and it can destroy your immune system, which we know now is very important in the battle against the cancer. So now we want to test whether we could use these focal adhesion kinase inhibitors in vivo. And so we went back to our zebrafish model, where in the mutant case that was very similar to the pancreatic cancer, where we lack sphingosine phosphate 2, you get these masses forming. Now when you add focal adhesion kinase inhibitors, you see these masses just fall off. So they're not present anymore, but if we film them, you can actually watch them fall away. Another important activity that this had was to restore the barrier function. Now, one thing I haven't mentioned is that when you disrupt cell extrusion, in this case with the mutant, that some cells still die, but they don't get extruded. And this can cause holes in that barrier function. And that can be seen when you add fluorescent dextrans. They just get taken up into the body because there's no very good barrier function here compared to the wild type case over here. Now, when you add focal adhesion kinase inhibitors, that also restores that barrier function in ways we don't really understand now, but that's really quite exciting for us. Partly because this inflammatory part of a lot of cancers can really help progress the cancers. So now what we see then is that we've identified this process for how epithelial cells die by this extrusion. And from identifying the signaling that drives extrusion, we found that when it's disrupted, cells no longer die, and they go on to form these masses that are actually chemotherapy resistant due to high survival signaling. Additionally, poor barrier function can result from cells that are dying in the epithelium but not extruding. And what we're excited about is that focal adhesion kinase inhibitors can reverse both of these impacts and restore these epithelia to normal wild-type behavior. So there are now clinical trials going on with focal adhesion kinase inhibitors and immune therapy for pancreatic cancer. So we'll be watching and hoping that this can help treat this awful disease. Now, finally, in this last part of my talk, I'll be telling you about another thing that can happen when apical extrusion is misregulated. And that is a sort of a sneakier type of extrusion, which is called basal extrusion. And we're wondering if that is a new way that cells can start to invade during metastasis. So while most of the time in the talk I've told you about cells extruding, I've really been talking about apical extrusion alone. So this is when cells contract around and below and squeeze the cell out into the lumen. That lumen is really dead space, so those cells can't go on to live. However, there's a small percentage of cells that go on to extrude basally, and that means that they extrude underneath that monolayer. Now here in cultured cells that you see here, this doesn't have that much impact, as in both cases the cells will die. But in the case in vivo, if cells extrude apically, even if they had high survival signaling, they would just go into empty space and be eliminated. But in the case of basal cell extrusion, these cells would go back into the tissue that the epithelial encases and could go on to invade into other parts should they be cancer-causing. So this idea was an interesting idea, but it was really made more exciting by the idea that we were finding lots of different oncogenic mutations that actually hijack this apical extrusion pathway and instead drive the extrusions to go basally underneath the tissue. And this was true in the case we just spoke about, when sphingosine phosphate 2 is gone in pancreatic cancer. And we're also finding that to be true in cases of colon cancer where there's APC mutations. So APC mutations are present in about 90% of colon cancer. And these also shift the direction of extrusion from apical to basal. And finally, what I'll be talking about for the last part is oncogenic KRAS mutations, which are known to be driver mutations in pancreatic, colon, and lung cancer. And so we wanted a way to test this whole model of whether basal extrusion could drive invasion. And so while most cancer studies have really occurred in mouse models, we needed a way to actually visualize every single cell that was migrating. And so to do that, we turned to our zebrafish model system because we can see every single cell that occurs. And we can also see cells developing tumors from the epithelium where they would actually grow and see how they would invade. And whereas in mice, we can't really see these things. So to do this, what we did is develop different drivers to express this oncogenic KRAS in different types of cells. So we have a driver now for the outer surface cells here, and also one for the basal surface cells here, which are the stem cells. But I'll really just be talking about driving this KRAS in the top outer layer. And when we do so, what you see is that these oncogenic KRAS cells form masses, very similar to the types of masses we saw with when sphingosine phosphate 2 is gone or piezo is lost, as you see here. Comparatively, epithelia that are expressing CAX, which is just a membrane tag GFP, which is inert, don't do this. You'll also notice that there's a lot more cells that survive at this outer surface layer here compared to the KRAS cells. And the reason for that, when you follow them in movies, is that these control cells will just sit on the top and not do very much. However, the oncogenic KRAS cells have a much higher propensity to extrude, and when they extrude basally for the most part. This can be seen when we quantify this. There's a lot more cells in each embryo that are extruding, and then most of them are extruding basally. However, a lot of these cells go on to die. So while some of these cells are alive, there's a lot of them that are dying inside the body. So we next wondered, because most cancers do not occur from just a single mutation, but really have multiple mutations, we wondered if a common collaborating mutation would help those cells survive after they had basally extruded. And so we put it into a background that lacked p53, so it was a loss of function mutation for p53. And p53 is a protein that's known to promote cell death, so loss of it would cause higher survival. And so when we do that, now you can see that these cells go on to survive, and they live inside, deep inside these, the body of the zebrafish. You can see in this rotating structure that the red cells here are the p63 or basal cells. So really, these green cells are deep inside of that body, and very few are now dying. Not only do they survive, but they go on to migrate, and you can see them migrating throughout the body here, as well as in here. So now we can really see all these single cells, and we really think that this is a good way to follow all this invasion going on. So together, what we found is that by just studying the basic cell biology, how epithelial cells die through this process of extrusion. And now we've found a number of oncogenic mutations that hijack this extrusion machinery to cause cells to not only form these masses and poor barrier function, but also cause cells to basally extrude. And we think that this basal extrusion could be a new paradigm for how cells initiate metastasis during tumor progression. Now, an important part of this is that these cells that are forming masses are very separate that we see than the cells that are extruding basally. And this is very different to the way that most people have thought about invasion. Usually they think that invasions happen from places of primary tumors, but we think that this can be occurring independently. And so this may change the way we start thinking about diagnosing cancers. Finally, this is an important part that we really need to target, because most people die not from the cancer, but because the cancer has metastasized. And this is where we've been really stuck a lot of times in cancer. is when people start to get metastasis, we run out of options. So now, because we can see these cells invading, we wonder if we can use targeted therapies based on what we know of how they should have died to target these cells now to die once they've already invaded. So I'd like to end by thanking the people who've contributed to all this work and also to my funding bodies. Thank you very much.